DNA replication. As we said at the beginning of this chapter that one of the features of a genetic molecule would have to be the ability to be copied perfectly many times over. It was not until 1953 that James Watson and Francis Crick used the result of work by Rosalind Franklin and others to work out the basic structure of the DNA molecule that we have just been looking at in figure 6.8. To them it was immediately obvious how this molecule could be copied perfectly time and time again. Watson and Crick suggested that the two strands of the DNA molecule could split apart. New nucleotides could then line up along each strand, opposite their appropriate partners, and join up to form complementary strands along each half of the original molecule. The new DNA molecules would be just like the old ones because each pair of strands could then wind up again into a double helix exactly like the original one. This idea proved correct. The process is shown in figure 6.9 on page 116. This method of copying is called semi-conservative replication because half of the original molecule is kept or conserved in each of the new molecules. The experimental evidence for the process of semi-conservative replication is described in box 6.1 on page 117. DNA replication takes place when the cell is not dividing. This is in interphase in eukaryotic cells. Most people thought that semi-conservative replication was most likely because they could see how it might work. However, to make sure, experiments were carried out by Matthew Meselson and Franklin Stiles in America. They used the bacterium Escherichia coli, E. coli for short, a common, usually harmless bacterium which lives in the human alimentary canal. They grew populations of the bacterium in food source that contains ammonium chloride as their source of nitrogen. The experiment relied on the variation in structure of the nitrogen atoms. All nitrogen atoms contain 7 protons, but the number of neutrons can vary. But nitrogen atoms have 7 neutrons, so most nitrogen atoms have 7 neutrons, so their relative atomic mass, the total mass of all the protons and neutrons in the atom, is 14. Some nitrogen atoms have 8 neutrons, so the relative atomic mass is 15. There are two forms. These two forms are said to be isotopes of nitrogen. We have learned in chemistry. The nitrogen atoms in the ammonium chloride that Meselson and Stiles supplied in the bacteria were heavy isotopes, nitrogen 15. The bacteria used the nitrogen 15 to make their DNA. They were left in ammonium chloride long enough for them to divide many times, so that nearly all of the DNA contained only nitrogen 15 atoms, not nitrogen 14. This DNA would be heavier than DNA containing nitrogen 14. Some of these bacteria were then transferred to a food source in which the nitrogen atoms all were nitrogen 14. Some were left there just long enough for their DNA to replicate once, about 50 minutes. Others were left long enough for the DNA to replicate 2, 3 or more times. DNA was then extracted from each group. The samples are placed into a solution of cesium chloride and spun in a centrifuge. The heavier the DNA was, the closer to the bottom of the tube it came to rest. Figure 6.11 shows the result of Meselson and Stiles experiment. Okay, so this is very easy. First of all, let me show you the process of DNA replication. We have seen the structure of DNA double helix. It's like this. These are hydrogen bonds. So first of all, these hydrogen bonds, this is a DNA double helix, this is an helical structure. First of all, the hydrogen bonds are broken. After the hydrogen bonds are broken, the DNA is no longer double stranded. Then what happens? Since we know that the bases can only form bond with their complementary bases. Oh yes, and in the human cell, the cytoplasm, there are a lot of free nucleotides being formed with these are free nucleotide being formed from a nitrogenous base, pentose sugar and phosphate. We have seen from where the phosphate group came and how this nitrogenous atom got into a chemical reaction with the pentose sugar. The both hydroxides made a reaction, glycosidic bond, and here also we have seen phosphodiester bond. So what happens? These free nucleotides having a specific base will line up along their specific base. Say, if this is an adenine and this is a cytosine, a thymine base will line up here and a guanine base will line up here. And by base I mean the nucleotide with the base will line up here, and also here the nucleotide with the base will line up here. But when it's a nucleotide, there are two phosphates. So then these phosphates are removed and they form bond with the nucleotides, and thus another complementary DNA strand is formed. Now let us see them in details from our book. But this is the basic. This is the DNA double strand breaks into a single strand. When one of the original strand is goes away, another the complementary nucleotides line up along the original strand and a new strand is formed. And they form chemical bonds and the new strand is formed, and this is the original strand. And so in every new DNA, the one original strand is present. So we say that one of the original strand has been conserved. Also for this strand there will be another new strand, and this is going to be the original strand. So this is a semi-conservative in a sense that one of the strand in the new DNA is the original strand. Let me show you in a more simplified diagram. Okay, these are double strands of DNA. This is the original. They split apart. Original. And complementary nucleotides line up and form bonds to form a new strand. Also here complementary to this original, and so one of the original strand is conserved, and so the one of the original strand is conserved and new strand. So this is semi-conservative DNA replication, and we have seen that from one DNA two DNAs, and one original DNA has been conserved in one original DNA strand has been conserved in the new DNAs. Okay, now let us see the process of replication in details from our book. This will be in figure 6.5. Okay, so a pair of nucleotide, each nucleotide is linked to the next by covalent bonds between the phosphate sugars. Pair of a DNA molecule, that is the number B structure. DNA molecule, two polynucleotides running in opposite direction are held together by hydrogen bonds between the bases. A link with the T by two hydrogen bonds C, like thymine and cytosine, links with G by three bonds, like adenine links with thymine by 2 bonds, hydrogen bonds, and cytosine links with guanine by 3 hydrogen bonds. This is complementary base pairing. Okay, so this is just a normal structure of the DNA. Let us see figure 6.9, which is the DNA replication. Figure 6.9. The DNA double helix winds and unzips as the hydrogen bonds between the bases break. And then in the nucleus, there are nucleotides to which 2 extra phosphate have been added. The extra phosphates activate the nucleotides, enabling them to take part in the following reaction. I'm sorry, in the diagram I showed altogether there are 2 phosphates, but in the nucleotide there are extra 2 phosphates added. So altogether there are 3 phosphates. So each of the bases of the activated nucleotides pairs up with its complementary base on each of the old DNA strands. An enzyme, DNA polymerase, links the sugar and innermost phosphate groups of next door nucleotides together. The 2 extra phosphates are broken off and released into the nucleus. So the 2 extra phosphates are released. There are 2 extra phosphates in the nucleotides along with the original phosphate. So what happens, I mean, like, how are the hydrogen bonds formed? I mean, like, how are the hydrogen bonds forms by itself, but how are the sugar and innermost phosphate groups of the next door nucleotides together hold? That is by the enzyme DNA polymerase. An enzyme DNA polymerase links the sugar and innermost phosphate groups of next door nucleotides together. The 2 extra phosphates are broken off and released to the nucleotides. So the nucleotides are joined together by the enzyme DNA polymerase. So DNA polymerase links the nucleotides together to form the new strand, which is complementary to the original strand. Then DNA polymerase will only link an incoming nucleotide to the growing new chain if it is complementary to the base of the old chain. Thus, very few mistakes are made, perhaps around 1 in every 10 to the power 8 base pairs. So the DNA polymerase links the nucleotides together to form the new strand of DNA, but it doesn't just link it like randomly. If the nucleotide is complementary to the old strand, then DNA polymerase will link the nucleotides to form the new strand, and then the hydrogen bonds are formed when the base pairs interact with each other. So now about the Meselson and Stiles experiment. Okay, so this will be in figure 6.11. Okay, so protein synthesis. No. So let's see. Say first of all, in a test tube, we have all nitrogen 15, which is heavy, and we introduce culture of E. coli. So after each replication, where new nitrogenous bases are needed, so after the E. coli replicates several times, all of the E. coli will have nitrogen 15. So after that, if we all of the E. coli containing nitrogen 15 from this culture is introduced to another medium containing nitrogen 14. What will happen? Imagine, in each DNA replication, 50% of the new strand will have nitrogen 14 because 50% of the new DNA molecule will contain the old strand, and the other 50% of the new DNA molecule will contain this new strand produced from nitrogen 15. So after one replication, 50% of the bacteria will contain nitrogen 14 and 50% of the bacteria will contain nitrogen 15. 50% of the bacteria will contain nitrogen 15. This is, and then after another reaction, that is another generation, after this generation have like what shall I say, after this generation have replicated another, we will have from here, once this 25% having nitrogen 14, having nitrogen, I'm sorry, not 25%, like, okay, after this %, will have nitrogen 15. After another replication, 25% nitrogen 15 and 75% nitrogen 14. Here is 50% nitrogen 14. So at every generation, since the culture contains nitrogen, medium contains nitrogen 14, at every generation 50% of the new strand will contain the nitrogen 14. So first of all, 100% contained nitrogen 15, but after replication 50% contained nitrogen 15. After another successive replication, 25% contained nitrogen 15, and like this, so on. So now, how did they find out that that the bacteria contains nitrogen 15 or nitrogen 14? Simple. There is a machine called a centrifuge machine, and in that machine, if you put the test tube, and inside the test tube we put a solution of calcium chloride. Okay, let's see here. Solution of cesium chloride, and then spun it in a centrifuge. That is, the centrifuge will like rotate it. That is, this is the centrifuge machine like this, and here we will put the test tube, of course with the lid closed, and this will like make it go round and round. What will happen? The bacteria that contains nitrogen 15 will settle, the heavier isotopes will settle at the bottom, and the bacteria that contains lighter isotopes will settle at the top. So we will see that after when we first introduce all of the bacteria with nitrogen 15, the bacteria settled here. After that, when 50% was nitrogen 15, it settled here. After that, when 25% was nitrogen 15, it settled here. So that's because the weight of the bacterium was decreasing as much percent contained nitrogen 14 and lower amount of nitrogen 15 was there. So well, there is a physics to it that why is in a centrifuge machine the heavier isotopes settle at the top. So there is a physics why the heavier isotopes like settle up and the lighter isotopes settle bottom the tube. I mean like settle above the tube, and why the heavier isotopes settle bottom the tube. Since this is like in a circular motion, we have a centripetal force. This is physics. And there is no such thing as a centrifugal force. So let's see the formula of centripetal force, that is motion of an object in acceleration of an object in circular motion, force of an object in circular motion. So omega equals to mv square by r. Omega is the centripetal acceleration. Mv square by r. So we have mv square equals to omega r, m equals to omega r by v square. This is centripetal acceleration. We know that F equals to ma. So where there is a force, there is an acceleration. And in centripetal force, the force is towards the center, then the acceleration is also towards the center. So higher the mass, higher the radius from the center. So it matched. This was our tube, right? This was our tube, and the centripetal force was this side, and this was in a circular motion like this, and this was the center, and we have a centripetal force towards the center, and higher the mass, higher the radiation. So we said that here we had nitrogen 15. So the radius, higher the mass, higher the radius. So the radius from the center was higher. Okay, so that's all about Meselson and Stiles experiment. It proved that DNA was semi-conservative because we know that at each replication the DNA doubles, and that we have seen that the number of nitrogen 15 at each replication was 50%. So it was semi-conservative. Now think in your mind about it and you'll understand it even more. This you do not need for your biology, this is for your physics A levels. I just showed you. Okay, so in this video I want to give a thought with you all. This thought was given to me by my Cambridge physics teacher in my O levels and A levels. So he showed us the logic that there is no such thing as a centrifugal force. Now when an object is in circular motion, say this is the object and this is in circular motion, this is the center. We said that there is always a centripetal force. And thus there is a centripetal acceleration towards the center. Now many people agree that there is a centrifugal force which is opposite the direction of the centripetal force and in same magnitude. But that is by logic wrong, because first of all, if there was a centrifugal force opposite the centripetal force in the same magnitude, then both the forces would cancel out. If the forces would cancel out, there would be no acceleration, because we know the formula F equals to ma. Since there are no forces and no acceleration, the body will move in a single straight linear direction with constant velocity or even constant speed. This is also by Newton's law that an object moves in a specific direction if no forces are applied to it. Does that make sense to you? Well, it did to me.